today we're going to discuss about the kinetics of cell growth in bioreactors. This could be interesting in tissue engineering or also in environmental engineering. So let's think of this kinetics of cell growth in bioreactors. Or in an environment, we can generally think. First, let's consider an actively dividing cells. Actively dividing cells. Then the rate of growth will be proportional to number of cells, assuming that there are all necessary environmental factors and nutrients are available. So the rate of cellular growth, the cell numbers will be proportional to the current number of cells. So how can I describe it? Like dn dt is proportional to n. And let's put the proportionality constant as a mu. So here, so there the n, it means total number of cells. And the mu, we can see this is a rate constant for cell growth, so or growth rate. So that means a rate constant for cell proliferation. So then you can solve this equation. So when you solve this differential equation, the number of cells will grow exponentially. With time. So that you can see those who can differentiate and becomes oneself into an exponential. So you can write it n equals n0 e to the mu t. Let's put it as this. So here I define n sub 0 is the time when time 0, the number of cells. So that is initial number of cells. Okay, then here we can actually consider the way when we are culturing cells, we know the cells grow pretty fast, like exponentially. And so that we can have defined the time when does the cell become twice. So we can call it as a doubling time. So doubling time for cell population as when the n becomes a twice. So that let's define the time as t sub d as a doubling time. That's when the cell becomes twice of the original, and then that would be n0 e to the mu t sub d. And so that we can solve this equation. And what is that? That means a ln2 equals mu times t sub d, or we can write down the doubling time as a ln2 over mu. So that, as we can put the equation 3. So here we'll see. So this is an ideal scenario, but in reality, the real cell growth, especially the cells derived from an animal, will be limited in terms of they have a special property of anchorage dependent and also density dependent cell growth. So first, let me write down what this anchorage dependent. So this is really when the cells are growing on a surface, when they contact each other or cells are crowded, their rate of growth will not be as, will be decreased. So that's because cell adhesion and spreading on a solid surface are required for their proliferation and proper function. Another thing, so the cells and spreading on a surface, growth on a surface is required. And another thing is the cell growth, when they are crowded, they have some contact inhibition property, so that this the cell growth is depending on really cell density. So that we call density dependent cell growth. So that is an influence of a substrate. Substrate, I mean the amount of nutrient or the surface environment for the cell to grow. So, or nutrient that limits cell growth. I mean cell growth, I mean cellular cell growth nature. So from this idea theory, we can move on to a little more realistic aspect. So that comes from so-called famous Monarch equation, who got Nobel Prize from his work. So that I will write down as a Monarch model or Monarch model. Okay. So this Monarch model is about really considering this influence of a substrate and the nutrients. So that limits growth. So this one is the leading to the Monarch model. And let me write down how does the Monarch model look like. So this is the from, if you look at from 1, so the growth rate is really 1/n dn dt. So that's by definition. And here we consider a little bit more from here by defining the mu max. So maximum growth rate, and then also consider the concentration of the substrate. Let's put as a bracket S. And also I introduce a parameter for K sub S. So this is the Monarch model. And let's put as a equation number 4. Here, let me define this here mu sub max is really maximum growth rate. Maximum growth rate. And of course, the S is substrate concentration. Then let's see. Then another one is K sub S is defined as half velocity constant. So half velocity constant. So what do I mean by half velocity constant? In fact, this one is when the substrate concentration is the same as this parameter K sub S, the growth rate will be half of the maximum. So that's how the name comes from. So when the substrate constant, when u over mu sub x becomes 1/2. Okay, so here really monad equation, let me say, so here mu max and k sub s is really empirical, empirical, uh, uh, empirical parameter. So that is depending on, so it's a function of species. So what kind of microorganisms and environment such as temperature, pH, and composition of a media. So let's consider this, look at this monad equation behavior a little bit more. So let me rewrite mu equals mu max times substrate concentration, Ks plus substrate concentration. So their independent parameter at the substrate concentration, we want to figure out how the growth rate will change. So first of all, like, what if there's no substrate? Then probably it won't grow. Let's check. When substrate concentration goes 0, then this 4 equation mu, the growth rate also goes to 0 unless the Ks is non-zero. So that's right. And then what if there's enough of substrate concentration? So then means we can mathematically consider there's infinity of a substrate. Then from equation 4, you can see mu goes to mu max, and that makes sense. So if you have a plenty of substrate, then probably your growth rate will be maximum. Then what about in the middle? So you can actually see like when substrate concentration becomes the same as this parameter k sub s, then you can see from the equation that because these are the same, so mu becomes 1/2 of mu max. And you can see why this Ks is defined as half velocity constant. In fact, this velocity means like growth rate. So let's actually try to write down, draw how this growth rate look like from here. So from the x-axis as a substrate concentration, y-axis as a mu, which is a growth rate. Then from the equation, when equals 0 equals 0, and then you can find out there's a maximum growth rate, which is mu max. So when s goes to infinity. So when substrate concentration is, let's say, Ks, then you can find half of this. So that point, so you can see the graph will look like this. And so of course, it's asymptotically closer to this one. So on another aspect of analyzing this is the initial growth rate change with respect to the substrate. So it's an initial slope. So when Ks is 0, so what the initial slope, we can consider this from the calculus point of view. So you can do this differentiation d mu over ds when substrate concentration 0. When you do this calculation, you will find out mu max over k sub s. So what that means is the initial slope of this curve is getting bigger when the maximum proliferation rate high and k sub s is low. So with that, you can actually figure out how the realistic cellular proliferation or microorganism growth in a tissue engineering when you need to grow extracorporeally from the patient's own cells, how well and how much you can actually grow to be able to implant to the patient. Or in environmental engineering, you can consider how microorganisms such as maybe useful bacteria or harmful bacteria, how they grow in terms of their kinetic aspects. And thank you for your attention.